And then in January, we're doing our group calls, and then later in January, we're doing another session on statistics. And then in February, I'm really excited, we have one of my favorite people, Paula, who's going to come on and talk to us about grant writing. She's been doing grant writing classes here at NIH for the fellows and is an outside institution who's going to come in and talk about how she's been successful with helping people get early career grants. So we're going to let Dr. Austin take back his seat. And here we go. No, they're going to be able to share the screen, and they're going to see your screen.
Good.
So here you go. And I'm going to be right over here.
Thanks.
As always, text me or put in questions, and I'll keep track of them.
Okay. All right. Well, welcome, everybody. I'm sorry I can't see you, but I suppose this is the future of education, that we get to go out into cyberspace. So I'm going to imagine you sitting here in the office with me, and we do have two people who are here, so I'll imagine I'm talking to them. But I'm hoping to give you at least one view of the translational problem, specifically as it applies to rare diseases. But much of what I'm going to say applies to all diseases, and I'll make a few comments about what I see as the convergence of common diseases and rare diseases that's happening in front of our eyes as well. So let's just dive in here. So sometimes people ask me why NCATS was formed as a new center at the NIH almost exactly 5 years ago now. That is translation a new problem? And of course, it's not a new problem, but I think it is fair to say that the level of opportunity is far greater than it has ever been. And as a result, the translational problem has become more acute because there are more things to translate, and therefore more patients who might benefit if we could get better at this. But we got a long way to go. And all of you who are on the phone are going to help with that. So thank you for joining the movement here. By the way, should I put this on full screen, or does it work just as it is?
You can if you want.
But it doesn't matter?
Doesn't matter.
Okay, I'll just leave it like this. So this really is the problem that we live in, or the world that we live in. It's an almost painfully bittersweet time where we know more about ourselves and health and disease than we ever have, perhaps exemplified by the Genome Project on the left there. But if you ask, has health improved to the epical degree that science has advanced in the last 30 years? The answer to that has to be no. This is a particularly pithy title from an IOM report a few years ago called Shorter Lives, Poorer Health, sort of says it all. And a fundamental problem here is that though fundamental science is unprecedentedly advanced, we are quite bad at going from what's on the left side to what's on the right side. That is, transition of a basic or a clinical observation into an intervention that is shown to improve human health. And there are all sorts of knock-on effects of this, and they're listed on the slides here. But the bottom line in red at the bottom is that people are unhealthier than they could be or perhaps should be. And funders of the biomedical research enterprise, you should know, if you don't know already, have become impatient with us in the last 10 or 15 years. And this is the public sector, the Congress and the taxpayers, and the private sector as well. But it's not all bad news. There's a lot of good news here, which also gets at the opportunity. That is, this is a graph I'm going to show you in a second of the number of human conditions that have a known molecular basis. This is data from OMIM. And when I graduated from medical school in the mid-'80s, that number was around a dozen, so it was a lot easier to pass your boards back then because there were only about a dozen disorders, the molecular basis of which you had to understand. Of course, we still had to know all of the crazy syndromes that you all probably have to memorize as well. But what's happened in the ensuing 30 years, is this, and I'm going to have to put this on here because the animation won't work. This is what's happened in the ensuing 30 years. And if you're asking why this knee in the inflection curve happened, it's because of the advent in the mid-1990s of large amounts of sequence data being put into public databases as part of the sequence phase of the Human Genome Project. And with those data, there are now over 6,000 human conditions. Most of them are rare, the molecular basis of which is understood. And that's wonderful news, but from a patient perspective, of course, what they want is not only a treatment and a pathogenesis, but they want a treatment. And on that score, we're not doing so well. If you look at all 7,000 or so diseases that affect the human family, only about 500 of them have any treatment. The rare disease numbers are a little bit more secure on the number of therapies level because of the Office of Orphan Products at FDA. So about 6,000 rare diseases is thought to be about that number, and only about 250 have any treatment that's approved by the FDA. So the patients who I used to see in the clinic, I'm a neurologist by training, and most of those disorders that I saw were rare diseases and were untreatable 30 years ago and are still untreatable 30 years from currently today. But you might say, well, gosh, you know, we all know, or you may know, that it takes 15 or 20 years to develop a drug currently from a novel target. So maybe this is just a time lag. Maybe if we just wait a little bit, there will be a flood of new therapies for rare diseases, but unfortunately you would be wrong. So I'm going to make an analogy here to Moore's Law. I hope you know what Moore's Law is. It's the reason that you can fit what was a supercomputer 30 years ago into your iPhone. That's this observation from Gordon Moore, who was one of the founders of Intel, that the number of transistors you could fit in a microprocessor has gone up linearly and the cost has gone down linearly over the last 40 years or so. And that has driven a lot of the technological miracles that we see all around us. But during the same period of time, therapeutic development has gone the other way. It's somewhat apocryphally known as Eroom's Law. This is Moore's Law spelled backwards. If you haven't read this paper, I would recommend it to you. It's sobering at best, truly frightening at worst. What you're looking at here is what in economic terms or productivity terms is known as dramatically negative, monotonically negative productivity growth in our ability or our accomplishment of making new therapeutics over the last 60 years. That is, that the number of new drugs approved per $1 billion spent has gone down by half every 9 years since 1950. That's quite extraordinary, and if you imagine any industry, any enterprise whose productivity consistently goes down by half every 9 or 10 years over a 60-year period, what would happen? That enterprise would, a lot of the organizations would go out of business, they would merge, they would acquire each other, and the products which are made would be exorbitantly expensive. I just described for you the pharmaceutical industry, particularly when it comes to rare disease therapeutics, that, as you may know, are typically priced in the $400,000 to $500,000 per year for the life of the patient range. So if you ask why this is, it's not surprising. The science would indicate that that's exactly what should happen, given the productivity problem. So the question is, how do we reverse this? Now, before I go on to how we're trying to reverse this, you should know, and those of you who are in training are going to have to deal with this with ways that oldsters like me are not going to have to. If you're asking when this line approaches zero, can't go to zero because it's a log plot, but it becomes truly asymptotic in about 2070. So formally, as of 2070, there will be no more drugs, or in other words, the cost of a new drug will truly be infinity. So it is an urgent priority to reverse this. But I show you this not to scare you, but to realize the problem that we were up against, and the fact that the invention of computers and CT scanners and mRNA and MR scanners and recombinant DNA and gene therapy, the Genome Project, none of those things have reversed this monotonic decline. Some people think it's actually made it worse. So NCATS was formed in part to bend this curve, because that's absolutely required if we're going to deliver on the promise of science for rare disease patients. And our thesis here is that this is a scientific problem and needs to be addressed in a scientific problem. The translational process has never been studied as a scientific problem before. It's never been approached that way. The translational process is almost relentlessly empirical from beginning to end. And when you have a process over this number of years with this number of increases in degrees of freedom as you go from a protein in a tube to a person in a community, if you have a trial and error approach with that kind of problem, the likelihood of you getting successfully from the beginning to the end approaches zero, and that's exactly what you see. But the answer to this is not more shots on goal, it's to understand the process. So that's why NCATS was formed. So NCATS's job is to allow interventions to be developed more quickly and deployed more quickly by creating new ways of doing things, new methods and technologies that will enhance this process, the effectiveness and efficiency of the translational process across all human diseases and conditions. And whenever I say all human conditions and diseases, this does include common diseases, of course, but numerically, most diseases are, of course, rare diseases. Okay, so the first question you have to ask yourself here is, how are we defining translation? This has become a quite in vogue term in the last few years, partially because of NCATS, I suppose. When I took over as director, we spent some months coming up with our definition. It's a quite expansive definition. It's the whole process by which an observation that a scientist makes in the lab, a doc makes in the clinic, or a community health worker in the community makes, a whole process from when that light bulb goes off and the person says, this observation in front of me is telling me something really important about this process, and if I can intervene to reverse it, I will address the target dysfunction, the disease, or the community health problem that's in front of me. So a whole process from which from the time that light bulb goes off to creating the intervention, the physical thing, the drug, the device, the behavioral intervention, whatever it might be, showing that it's useful in individual people and controlled environments of clinical trials, and then in the community, because that's where health happens. And our job is not done until we've had the effect on public health that we thought we were going to have when the light bulb went off at the beginning. Good. So that's translation. But you notice that NCATS is not the National Center for Translation. Virtually every center and institute at NIH does translation, at least some of it, according to this diagnosis, or this definition. And every biotech and pharma does. We're the National Center for Advancing Translational Science. So the first question is, what is translational science? It's a young field, which is one of the reasons it's so exciting. It's a young field that's just being developed now, and you are at the vanguard. It's the field of investigation that seeks to understand what the general principles are, scientifically and operationally, that underlie the translational process, with the thesis that if we understand those general principles, this process will go from being an empirical, phenomenological process to a predictive science. That's happened in every other area of science, and we firmly believe that it will happen here as well. But what's a little unusual here is that this is a scientific problem as well as an organizational problem. If you ask why the translational projects fail, about 50% of the time it's because we don't understand the science, but about 50% of the time it's not hard science. It's not biology, chemistry, physics, or engineering. It's intellectual property. It's incentives. It's credit. It's bioethics. It's something which is not strictly in the realm of what many of us were taught science is. And so because those are translational roadblocks, NCATS views these as areas that we need to work on as well. So we innovate in all those spaces as well. Okay, so sometimes people ask me, okay, I understand this now, I understand what you all are up to, but what do you all actually do? What are the problems that you work on? And this is a partial list. I think a lot of you are docs, and if you're a doctor, you know that you're taught to keep a problem list for all of your patients, and this is the translational patient's problem list. And it's the first three are the big three reasons that drugs fail before they get into people. The middle ones span the preclinical to clinical domain. The ones at the bottom are more clinical. But the important thing here is that you don't see a disease name in any of these. And that's because it doesn't matter whether you're working on Menkes disease or DMD or epidermolysis bullosa. It doesn't matter what the system is you're working on, or you're working on Alzheimer's disease for that matter. These are all the same problems, no matter what the disease is. So they fit what economists would talk about as the tragedy of the commons, and this helps you understand why these problems exist in the first place. The tragedy of the commons is a well-known economic principle, which is that problems which are everyone's problem in general are nobody's problem in specific, so it's nobody's job to fix them. So that's been the case up until NCATS has been formed, because at the NIH we have institutes focused on individual organs or individual diseases. We have doctors focused on individual organs, individual diseases. We have companies focused on individual technologies. We have foundations focused on individual diseases. There's nobody whose day job it was to solve these problems. So everybody over the last many decades has been trying to manage with a system that is fundamentally flawed, grossly inefficient, because they're just trying to develop a therapeutic for their individual disease or their technology, which is perfectly fine, but it's led to the mess we're currently in. So that's the attitude that NCATS has is a little bit different from what you might be familiar with. Okay, so how do we actually do what we do? That is actually a little bit more conventional. I'm not going to go through all of these. These are all actually some of the programs that NCATS has, but the point I want to make from this slide is that they fit into three rather conventional bins. There's the clinical bin, which is about 75% of what we do. There's the preclinical bin, which is about 15% of what we do, and broadly re-engineering the translational process, which is maybe about 10%. And I'm going to just show you a few examples, or maybe one example from each one. Okay, so the first is not a rare disease specific program, but it gets at one of these general enabling, solve the overall problem programs, which will disproportionately benefit rare diseases, because rare diseases are most abundant and in many ways in most medical need, as you all know. So what is the CTSA program? It's actually the NIH's biggest single program. Many of you are probably at institutions, academic health centers or medical centers that have CTSAs in them. It's a national consortium of medical research institutions that work together on the sort of bottom two-thirds of that problem list I showed you a minute ago. Importantly, provides innovative training for clinical translational researchers, which is a little bit different from how conventional training is done. I'll come back to that a little bit. I don't have time to go into all the aspects of this program because it's a very large and diverse program, but it's important for you to know that one of the things we started working on soon after I became director in 2012 was something which we think it will really revolutionize how we study novel interventions in human disease. That is that we currently have a terribly inefficient atomized system for doing clinical studies in this country for a variety of reasons that we could talk about maybe if we have questions, if we have time for questions at the end. But the important thing is that from a patient perspective, this is really limiting to the development of understanding of human diseases and treatments for those diseases. Clinical studies in humans, studies in human subjects, are inefficient at limiting the translation of basic discoveries and interventions that improve human health. And so we started about 3 years ago, almost 4 years ago now, focusing on three of the limiting steps in this process. If you think about this as it's a bad analogy perhaps, but it's useful for this purpose. Think about what we're talking about as a biosynthetic pathway, and you are interested in what the limiting steps in the biosynthetic pathway are, and developing catalysts to get over those limiting steps. Those were multi-site IRB review, recruitment to clinical trials, and duplicative investigator qualification. There are other things we've since worked on, like budgeting and sharing of electronic health records. But the important thing is that we've solved a number of these original problems via a trial innovation network that's going to be launched shortly, which is what we've called at the top, and each of these words is chosen on purpose, an efficient, collaborative, national network for translational medicine. Now, that does not exist now. It's not efficient, it's not collaborative, it's not national, it's not a network, and for many reasons, for many ways, it doesn't do translational medicine either. So this, we think, will really transform all the work that all of you are doing. The CTSA sites are all these places. Some of you, or many of you, are probably at these point sources, but the important thing is that these CTSA sites are not really, they are point sources, but they really are, there are many local collaboratives which have been put together over the last few years. And what we're doing essentially here is to create a national network out of these. And some of the benefits might be obvious, but they are to be able to form virtual teams to be able to do just-in-time clinical studies. It's particularly important for rare diseases given their rarity, and not only the rarity of the patients, but the rarity of the investigators. Sharing information, practices, tools across these 60-odd places. There is amazing accomplishment in each of these places, but up until recently, they really have not shared with each other. They haven't been incentivized to do that. Connect data systems to be able to connect patients and researchers, implement efficient multi-site studies. Importantly, integrate care and research. You may or may not have realized that we have a quite odd situation in this country where in many, many places, and as a rule, the care of patients is parallel to the research on the diseases that those patients have. And in many places, literally, the health center is across the street from the medical school and the research center, but often very little cross-pollination happens. There's often independent governance, and this is really a crazy system for patients and for translation, and it impedes what we would all like, which is something called a learning healthcare system or a learning research care system. And so we're really uniquely positioned within the CTSAs to fix that problem as well. And of course, something that will come up over and over as in my remarks today is that whenever I look at why we are more successful, even over the last 4 or 5 years, at solving some really difficult problems, the secret sauce is always the same, and that is that teamwork is the answer to almost all problems we have confronted. That is, if you take expert individuals, you put them on a team, and you give them a really hard problem, we have yet to find a problem that is not solvable. The problem has been that our system often prevents people from effectively working together. But when we can change the system to allow that to happen and align the incentives, really magic happens, and I hope you're being trained in that kind of ethos, because that's really the way of the future. To that end, within the CTSA program, we do a lot of research training, but it's really not your father's training or your mother's training. This is innovative, tailored curricula for including non-traditional areas like regulatory science, entrepreneurship. We're in the course of setting up required externships in industry, foundations, FDA, et cetera, on the concept here that if we're going to develop translational scientists, we have to think about them the same way as we think about translational linguists. If we're going to train people in a novel language, we don't just sit them in a classroom. We teach them the language in the classroom, then we send them abroad to study in that culture, and they learn the culture and the word use and the vernacular and the history and all that, and then they can be good translationists. The same thing is true on the science level. And the bottom line here is that we're creating an environment in which translational research is not only a viable career plan, path. It's an attractive career plan, and over the long term, creating an academic discipline of translational science, which has all the same panache and as genetics and biochemistry, is really going to be important for all of us, and hopefully this will happen in my lifetime, not just yours. Okay, so before I get into some preclinical stuff and some specific rare disease things, I want to just take a little bit of a second here, a few minutes, to help you think about what you hear a lot, which is this new term of precision medicine, which is pretty much the same as what used to be personalized medicine, but it's now been rebranded precision medicine. And the reason I mention it in this context is that precision medicine is the kind of medicine that rare disease docs have been practicing for years. So there is very little under the sun that the precision medicine folks are confronting that the rare disease docs and researchers have not already been working in and used to. And so I think there's many lessons that the precision medicine people need to learn from you all and the rare disease world in general. Okay, so why is this happening? It's happening because if you look at prevalence on the left side, we have a continuum now. There's what's been called common disease, and for common diseases, or heart disease, what used to be considered big cancers, Alzheimer's disease, these kinds of things. I'm going to use the converse of the rare disease definition to define a common disease. That is, if it's over 200,000 prevalence, that's a common disease. A rare disease, defined by the FDA, is less than 200,000 U.S. prevalence. And then we have personal diseases now. These are N of 1. These are becoming quite common now, not only through the Undiagnosed Diseases program that I'm sure you're hearing about in this course as well, but also through things like cancer immunotherapy, which are directed at the individual cancer of the individual. So these are really personal diseases. So what happens in the top is that what were previously common diseases, things like all lung cancer, are being segmented into smaller collections, molecularly defined, which fit the rare disease definition. So everybody's favorite example is all lung cancer, which is at least my favorite example. All lung cancer is about 200,000 new cases a year. Lung cancer that has a particular marker called ALK, that's about 5,000 U.S. prevalence every year, so firmly within the rare disease world, about the same as Hunter syndrome. I just took a random example of a rare disease, but there's hundreds of others, thousands of others, about 2,000 U.S. prevalence. And then there's individual diseases, things like CAR T cancer therapy, and one of my favorite diseases, which is NGLI1 disorders, which was a N of 1 just a few years ago. And the name of the game on the bottom is actually to find more patients. Start with an N of 1 and see how many patients in the world you can find via international consortia, national consortia. Whereas on the top, what you're trying to do is find a common disease and segment it. There's little doubt that virtually all diseases will end up being rare diseases within a fairly short period of time. There are some disorders, mainly the neuropsychiatric ones, which are going to be resistant to this, Alzheimer's, schizophrenia, bipolar, because they've been very difficult to define molecularly. But this is clearly already happening in cancer, and I think will happen in other diseases as well. Now, the societal problem here is the cost on the right. So as you might expect, as the prevalence goes down, the cost goes up, because the cost of developing a new therapy is about the same, whether it's for 10 people or 1 million people. And so if you have fewer people to get your return on investment, you got to take the prevalence and multiply the cost by 1 over the prevalence. So that's one of the reasons why the costs have gone up as much as they have, and you see that problem there. Okay, so with that in mind, let's now talk about rare diseases. So at NCATS, we are the epicenter for rare diseases at NIH for a variety of reasons. We have a lot of different programs, the RDCRN, which you all know about. It's currently 22 consortia studying over 200 diseases, this Genetic and Rare Disease Information Center, which is a source for information for parents and their patients and their families, important scientific conferences program that define new research agendas for novel rare diseases in this global rare disease registry. I'm not going to have much time to talk about, but a big issue in rare diseases is registries and natural history studies. I know many of you are working on these issues. This is the world that probably many of you live in, the Rare Disease Clinical Research Network. I just want to make 2 points here of how you may not realize how unconventional this network is, and it's really 2 of the reasons why this program has been outsized productive compared to other initiatives of its size. And they're both featured here. One is the requirement of the PAG. That's a patient advocacy group. So in order to get funded, you have to have a patient advocacy group, at least one as a member of the research team. That's becoming quite common now, but when RDCRN started, it was very unusual, even unprecedented. This gives urgency and focus to the research team, which really is irreplaceable in my experience. The other is that we required that the PIs not focus on individual diseases, which they'd previously done, but to give us a level of biological organization that they are going to use to study multiple rare diseases. So the model here is that we are going from thinking about individual diseases as individual entities unrelated to all the other rare diseases, to the reality, which is that these are 6,000 pieces in a very complicated jigsaw puzzle, but all these pieces are related to each other, and they need to be studied and approached in that way, so that as each puzzle piece is put in place, the number of possible positions for the next puzzle piece goes down. And so what was done here was to tell people, we don't care what your level of biological organization is, tell us how are you going to group rare diseases. And you notice some people did what Marshall Summer did and say, I'm going to study everything in the urea cycle. Some people decided that they're going to study an organelle, like a mitochondrion or a lysosome. Some people say, okay, I'm going to study all diseases that affect a cell type, like the peripheral nerve. Some people were very ambitious, and they took on a whole organ, rare diseases, rare lung diseases. And each of these has been outsized productive because the insights in one disease get rapidly applied in others in the same category. Something we started relatively recently we're really excited about, which is to help patients be more effective partners with all of us scientist types, to provide a patient toolkit. We've all, I'm sure, talked to patients and parents who are bewildered when they get a new diagnosis and they want to know how they can help. And they often feel like they have to recreate the wheel because there are not resources out there. They don't know how to find them other than just Googling something, which is frequently not terribly effective. So what this will do is provide a central web portal to online tools and resources that patient groups can use across the drug development, therapeutic development process. And these will really be from a how-to perspective. How do I do this? How do I establish a patient registry, et cetera? So you can think about it sort of as current protocols, if you want to think of it that way, but for a patient group. So this will be out with a beta of this. Currently, the schedule's in March, so be on the lookout. I want to just use the Rare Lung Disease Consortium as an example here for reasons that will become obvious. As it says here, it's a network of physicians and patients. Notice those are together, working to accelerate clinical research and improve patient care. It's run by Bruce Trapnell, who's at Cincinnati, but there's many other investigators in this consortium. And these are the 3 disorders that they currently study: lymphangiomyomatosis, pulmonary alveolar proteinosis, and Hermansky-Pudlak syndrome. And one of the things that this consortium did is develop the first FDA-approved drug for the first disease in this list, lymphangiomyomatosis, which among other things, if you want to impress your friends at a cocktail party, if you can say that word, you will impress all kinds of people. So You might want to take that home and practice. But LAM is actually a horrific disease. It is a rare progressive lung disease, mainly of women of childbearing age, which leads to gradual destruction of the lung, and these women generally asphyxiate because their lungs cease to function. And there is no treatment, at least was no treatment before Frank McCormick, who is the PI of this center, well, co-PI on this center at University of Cincinnati, set up a collaborative effort between this rare disease consortium, Pfizer, who makes the drug that was tested here in the LAM Foundation, Patient Foundation, to do a clinical trial of Sirolimus, which is a, well, I'll get to that in a second. Sirolimus as a potential treatment for LAM, and it was the first FDA-approved drug for LAM. So if you ask why this happened, it didn't happen randomly. It happened because there was a team that came together that put aside their own individual immediate gratification to focus on what the problem was that would benefit patients. So the drug company that made it, the patient foundation that wanted the treatment, and the academic investigators who were studying it. And what happened here is what happens, we see over and over and over again. When you do a program like this, the success is so much faster, so much more efficient, that there is more than enough credit to go around. So by putting aside the initial focus on how we're going to divide credit and working together for something that will benefit patients, then the academic goals, the company goals, and the patient goals are served better. And I know I must sound like Pollyanna here, but this is exactly our experience. So I hope you'll take that to the bank. Okay, so, but if you are like most rare disease patients, like the LAM patients before last year, most of them were in that big yellow bar, and that is not a happy place to be. So how do you develop a drug? And I'm not going to go through all this in any gory detail, but you just need to have this in your head if this is not something that you have done before, and I'm assuming most of you haven't, is that it's a complicated process, but you can boil it down to essentially 4 stages. One is the stage that many of us who are trained as basic researchers, which I am. I'm trained as a neurologist on the clinical side, but as a model organism neurodevelopmental geneticist on the basic side. So a basic research project is what starts many of these. A discovery is going from a target to an intervention. Usually that's a small molecule, could be a device, could be an aptamer, could be an siRNA, could be CRISPR these days. To develop a prototype takes you about $1 million. To go into early clinical, first in human, that's about $10 million per project. Late clinical is about $100 million per project. Keep in mind that there's about a 99% failure rate from the left side to the right side currently, which is why this is so expensive. It's not the unit cost, it's the failure rate. But what I want to bring your attention here is a really awful Sophie's Choice situation that people like me at NIH find ourselves in currently, is that there are many, many, many thousands of discoveries which can be translated and could benefit human health, but we are in a flat budget environment. And for every preclinical project that costs $10 million, I could fund 100 postdocs at the basic phase. So I'll just let that sit there for a moment. We have to make those decisions every day. Now that is not a comfortable situation for anybody to be in. I think the way to think about this is I firmly believe that if we can show to our funders, who are the taxpayers, that we can deliver on the therapeutic interventions, that more money will come to NIH, but it's not going to happen the other way around. But realize that there's a log increase in the cost per project as you go from beginning to end here. So just keep that in mind. Okay, so the NCATS preclinical innovation piece is designed with this obligatory team model in mind. That is, the idea here is that there are many academic investigators, mainly academics, some foundation and companies as well, who make discoveries, but they don't have either the inclination or the experience or the resources or the environment to get into true translation or therapeutic development. But there are people who really know drug development very well who may not know the rare disease very well. So these academic investigators get stuck at these various places at the top here. Those are those blue arrows at the top. And the idea here is that each of those people gets paired with a project team at NCATS who is made up of drug development experts that come from places like this. These are 3/4 of the folks at NCATS, this part of NCATS, come from the private sector, come from biotech and pharma, so they know how to do this stuff, but they wouldn't know lymphangiomyomatosis if it hit them in the head. But the LAM people don't know what pharmacodynamic interactions are either, which is a classic drug development problem. So the point here is that we form a joint project team, academic investigator who is the target disease expertise with the NCATS team that has the drug development expertise. They have milestones and go-no-go decisions, and they go into one or more of these programs here, and at the bottom come deliverables, and those deliverables move the project down the translational pipeline, and also, importantly, have a technology development component that help us learn how to do these better. So each project we get better at, the efficiency increases. So just one example here, don't worry too much about the details, but this happens to be on galactosemia, a rare disease many of you probably know about. There's 3 different forms. The therapeutic hypothesis here is that galactokinase, inhibition of galactokinase could prevent the toxic buildup of what is thought to be the bad actor here in one hypothesis, galactose 1-phosphate, and improve patient outcomes. That comes from an observation of the difference between classic galactosemia and type 2 galactosemia. And again, our people didn't know anything about galactosemia, but Kent Lai does. And so Kent came to us and said, can we work on testing this therapeutic hypothesis by developing inhibitors of galactokinase? And so we created a high-throughput screenable assay, went through about 300,000 compounds in 10 different concentrations, did a lot of med chem on these compounds, looked at cellular efficacy, then did all the PK/PD work, did co-crystals, and are now in animals. And the goal is then to move these into people. And we have many dozens of stories like this. And through each of these, we're not only developing proto-drugs for novel rare diseases, but also learning how to do this better, faster, cheaper. We do a lot of repurposing. I hope you know what that is, but if not, this process is very scientifically interesting, but is very long and difficult. Typically starts with around 500,000 compounds, takes about 15 years to go from the left side and the right side. So the idea that we and many other people had is if you took every drug ever approved for human use and you put it into a collection, screened it against human relevant assays, could you go into clinical trials in those patients in a relatively short period of time? When we started this about 7 or 8 years ago, it amazed us that this phrase I use so blithely, a complete non-redundant list of all of those 3,000 approved drugs, every drug ever approved for human use worldwide. One would think that that would have been done already. What we found in 2007, that had never been done. And you say, how is that possible? How have regulatory agencies never put together a complete non-redundant list of every drug ever approved for human use in the major markets, that is US, Europe, Canada, Japan? And the reason is it's a tragedy of the commons problem. It wasn't anybody's problem to do, so it had never been done. It took us about 5 years, but it's now published, and you can download the whole thing off our website. And the important thing is that there's now a physical collection as well, and we do a lot of collaborative screening of rare diseases to look for new potential treatments. And we do a lot of work with scientists, but we do a lot of work with patients, too. And here are 3 examples. These are 3 rare diseases, projects on Charcot-Marie tooth disease, on chordomas, rare cancer, and myotonic dystrophy. Each of these started because patients walked into my office and asked if they could work with us. And they nucleated the scientists, not the other way around. And this has turned out to be a very effective model. We find that the patients these days often know as much or more about their diseases than the scientists do. So that's been a very effective model for us. But let's say you get to this point, so you've got a GALK compound, or you've got a potentially repurposed compound. Most biotechs or pharmas will not pick it up at that stage. You've got to show safety and efficacy in a person. So we have a number of programs at NCATS Trend and Bridges focused on these trends, specifically on rare diseases, as the R in Trend goes through lead op through phase 2 clinical trials. Just want to show you one example here, which is going to take us back to the RDCRN. So these started completely independently, and they've shown the kind of convergence that happens when you create an ecosystem like we have within NCATS. So autoimmune PAP is a, unfortunately, typically horrific rare disease where these kids have circulating levels of neutralizing anti-GM-CSF autoantibodies, and that leads to abnormal surfactant development. So this surfactant builds up in the alveoli and is not cleared. So these kids literally fill up with surfactant, and the current treatment is whole lung lavage, which is just as bad as it sounds. What one does is to bring the child into the operating room. You put an endotracheal tube into one main bronchus. You anesthetize them, and then fill the other lung with saline, and you shake the kid up like a juice bottle, and you pound on the kid to try to get all of the surfactant out of his lungs, and you turn him upside down, and all the saline comes out with the surfactant, and you rinse and repeat, rinse and repeat, rinse and repeat, and you see on the bottom there eventually, gradually this clears. It is a horrific, barbaric process, but it keeps these kids alive. So Bruce Trapnell thought, well, gosh, you know, if it really is true that we have this autoantibody problem, GM-CSF was approved a number of years ago as a treatment for leukopenia, mainly in cancer treatment, but it would have to be delivered inhaled for this, and that the company Genzyme really didn't have any interest in doing that. So he came to us and he said, well, gosh, you know, I've got the company that's willing to supply the drug. I've got patients who desperately need this treatment, but we don't know anything about how to develop this. Can you help us? So NCATS acted as an adapter, as we often do. Got all the chronic GLP tox studies done, formulation, helped them submit the IND. That was cleared in the phase 1 study, was started earlier this year. So this, again, is why did this happen? It's teamwork. Teamwork. If Bruce had tried to do this on his own and learn how to make drugs or try to develop GM-CSF, this would never have happened. If Genzyme tried to do this without the experience of Bruce Trapnell, that never would have happened either. So, but together, we were able to make enormous progress very quickly. Okay, so lastly, I'm going to give you something which I hope will make you also excited about the future, which is not this slide, not why drugs fail, but what we're doing about why drugs fail. So another of the really quite shocking numbers that I'll give you today is that if one gets through that process which I showed you, getting into people spending tens or hundreds of millions of dollars, even when you get in a phase 1 study of a novel drug, let's take away repurposing for a second because that's a special case, but most novel drugs are novel composition of matter, as we say, a novel structure. 9 times out of 10, those drugs never make it to the market, even when they get into people. So if you hear all this celebration about going into a phase 1 trial with a new drug, don't believe it, because 90% of the time, it's never going to make it to patients. So why is that? You'll see here on the left side, about 55% of the time, it's because the drug looked like it worked in animal models but had no effect in the people, or about 30% of the time, it looked safe in the animals and had adverse effects in the people. So what you're seeing on the right side is data that shows you that about 50% of the time, animal data are and are not predictive of toxicity in humans. So it's about a coin flip. Efficacy is even worse. So this tells us what, when I mentioned this to my family at a holiday get-together a couple of years ago, I was very proud that we had discovered this, and the community had discovered this. And my family, of course, said, so let me get this straight. You have discovered that rats are not humans. Great. How much money did you spend discovering this? But when you think about it, that we have all been sort of brainwashed into this idea that these are animal models, an animal model for a disease. But a model is not the real thing. A mannequin is not a person, and a mouse is not a human. So we've learned this the hard way, and I've certainly learned it the hard way in my own career. So we're taking multiple approaches to this. It's not that animals can't tell us things. They can. But again, it's context of use. What are you using these for? And is it possible to develop these days more predictive systems which depend on human cells, or human systems, not animal systems? So one of the ones we're doing here is this program that we call the tissue chip program. The idea is to develop in vitro platforms that use human tissues to evaluate the safety and efficacy of promising treatments. The idea here is to utilize a commonly used word, or an increasingly commonly used word these days, which is convergence. It's the concept of taking disparate developments in science and engineering and medicine and putting them all together, and can you do more than you can with the sum of the parts? And so what we're bringing together here is stem cell technology, tissue printing technology, microfluidic technology, and cell sensor technology, putting them all together and asking, can we create human organoids, which represent structural and functional elements of all human tissues, that are more predictive of giving these potential therapies to an intact mouse? Now, we don't know the answer to that question, but I'm going to give you the 4.5-year progress report, and I must tell you it's very promising. So if you're interested in hearing more after this, you can go to our website. That gentleman on the right there it's actually not a gentleman, it's a gentle person, because he has both sexual systems, actually. But that individual is called Chip. Yes, he's on our website. And if you click on any of Chip's organs, it will take you to a microfluidic platform representing those organs. We spent the first couple of years developing each of the organs, and the last several years integrating these into a modular platform, getting all 10 organs in one platform. Importantly, this was a collaboration, like everything NCATS does, in this case with DARPA, which is the Defense Advanced Research Projects Agency, people who brought you the GPS and the Internet, and the FDA, and the current initiatives or current problems we're dealing with are integration of all these organs, testing of canonical compounds and then compounds in development, validation of these systems in external labs that we've just funded, and then partnerships to get adoption going. So just to give you a sense of how this works, and I hope you're thinking about this in your own disease, all of these have the same basic components. They all start with a scaffold of some sort, usually some sort of extracellular matrix on which are layered cells, and these are iPSC-derived cells or primary cells. They have a structure which either is given to them or they take it on themselves. Different organs are different. They often have to be perfused, because I should have said that all of these have to be viable for at least a month. Often most commonly they're viable for 3 to 4 months now. They have to be in bioreactors to keep them alive without being contaminated over that time. They often have to be innervated, right? Because if you have a gut that's not innervated, it's not going to work. Often have to have a host response, because many toxicities are due to immunological responses. Have to have a functional readout, which is non-destructive, so that you can go back and you can monitor this over the course of the months. And then you have to have a computational design that allow you to gather the data. And to make this happen, and I'm going to sound like a broken record here, this is a team. It's a very large team. And the coaches in this case, that are otherwise known as project managers, is the FDA and NIH mainly, but also FDA and DARPA, These are all scientists, PhD, MD level scientists who are managing this. Think about Bill Belichick in the Patriots. You know, makes all these, helps all these fantastic players work together in a football game. Same thing here. You know, fantastic researchers at all these different academic research institutions working on different organs. Then we have 2 engineering programs at the Wyss Institute at MIT. Who are working on engineering, and then a large number of biotech industry partnerships on the left side. And I'll just give you one example. This happens to be the liver, because it's kind of an easy one to understand. There's a bunch of different cell types, endothelial cells, Kupffer cells, stellate cells. You've got to have a collagen matrix on which these live, and then, of course, the hepatocytes. That's the schematic, but what this actually looks like in the chip platform on the left, and what it actually looks like under immuno-EM, immunofluorescence in the middle there. Looks pretty much like a liver sinusoid. An interesting observation here is that these systems really do behave like in vivo systems do. I'll just draw your attention to the lower right. This is the B panel. This is looking at the system under static co-culture systems, and you'll notice if you have that, static just means how I've been doing tissue culture my entire career, which is without flow, so that the media sits on top of the system, multicellular system, but the media just sits on top. In that case, the system does not produce the maturity, the markers of maturity, which in this case are albumin and urea. If you introduce flow going through the sinusoids, the differentiation, terminal differentiation happens. They begin to produce albumin and urea, just like happens in vivo. This is something we see over and over and over again, that the physical environment, the stretch, the flow, is critical to having a mature function as well as all the cell types. You can then introduce biosensors into each of these cell types to report out on various readouts that you might be interested in. This happens to be cytochrome C release upon addition of a toxicant there, if you see the movie, it follows exactly what you would expect upon the toxicant. I don't have time to go through all this. I just want you to get the sense of the red-green here. What you're looking at are canonical compounds. On the top is a compound many of you have probably used, trazodone, which has very rare, extremely rare liver injury. In this liver model, it gets all greens. If you go down to something like triglytazone, which is a PPAR agonist withdrawn from the market a few years ago, that's got some red and some green. And then a frankly toxic compound, metadione, gets mainly red. So where are we going here? Now that these chips have shown to really be able to produce in vivo kinds of responses in ways that have frankly overperformed compared to what I at least expected, and I think many people have. What we've now done is to create a whole program based on these chips to explore ways that they might be applications to which they might be put in addition to the initial use for testing safety and efficacy for drugs. And I put in red a couple of new programs that we've just started. Last fiscal year, we started a particular FOA on rare disease research rare disease modeling. We found that these can be very effective systems for developing rare disease models. And in FY17, we're expanding this to all diseases, still focused mainly on rare diseases, but focusing on disease pathogenesis and systems for testing interventions. But eventually, by eventually I mean in the next couple of years, what we're beginning to think about is on the right side applications in precision medicine. That is, if you think about these chips as being made from primary or iPS-derived lines or cells, it is as easy to make 100 different chips from one person as it is to make 100 different chips from 100 different people. And so then one can begin to think about testing novel therapeutics across a population of people for safety and efficacy. One can think about looking at all patients, I mean all in this case, of patients with a rare disease. There's often very broad heterogeneity, phenotypic heterogeneity in these patients. Make a chip out of all of those patients and see if you can understand why. Where does that heterogeneity come from? and doing it without making a transgenic mouse of every single one of those patients, which is just not scalable. So it's a pretty exciting world that you all are getting into. I must say I'm a little jealous of you. And if you're interested in hearing more, we just released a strategic plan a few weeks ago. It's on our website. It's more of a strategic vision than a strategic plan, but I think you'll capture both some of the exciting science, but also some of what we view as the real exciting potential of the field. So if you're interested in hearing more about us or connecting with us, we have many ways that you can do that on the web, Twitter, Facebook, et cetera, and I hope you'll take advantage of that. So thank you for your attention. I know we've probably gone late. If there's time for questions, I'm glad to take any. Otherwise, we can archive it and be done for the day.
Don't move here. Oh. So thank you. Now I think everyone who's listening online and everyone here knows why we're here in your office today. Exactly what we wanted to hear about, kind of your vision of where rare disease is going next. So thank you so much. We're just thrilled that you took time to do this with us. Thank you so much. I do have one texted in question, but if anyone has questions that you want to go ahead and hop on now and ask it out loud, of course, you can always text it or post it if you're too shy to say who you are. So I'll give just a couple seconds. And then
or you can just email me directly.
Yes, or you can email it to me and I'll send it to you.
Yes.
Either way.
Yes, when we say we're collaborative around here, we're not kidding. We actually want to hear from you.
This is one of the reasons why we want to make sure that everyone heard your voice and knows you and gets that invitation. So I do want to say we had one of the people that did the LAM work was in our group last year.
Ah,
so it's kind of fun to see all their work showing up on the screen. So
wonderful. Yeah, well, hopefully one of you who are listening today, I'll be able to feature you next year.
We'll put you next year.
Right, exactly.
So hurry up, guys. Yes.
That's right.
So the one question that was texted to me, and it was actually texted to me this morning before the session, and their question was, if you were to do it again, would you do rare disease again, or would you, like, where would you invest your time if you were starting over now in clinical?
Oh, boy.
Good question, right?
Yeah,
overwhelming?
What a great question.
Yeah, I thought so too. That's why I asked it.
Well, so let me answer that by giving you what my random walk was and why the answer is I would do rare diseases. That is that when I went from my academic position to industry, industry wasn't interested in rare diseases then, and because it's in the late 90s. And so I began working on schizophrenia and Alzheimer's disease, which are two horrific diseases. I think we're all familiar with that. However, if I look at how much progress we were able to make in those disorders, I'm quite disappointed, frankly. And if I look 20 years later, are we really any farther ahead on schizophrenia or even on Alzheimer's than we were 20 or 30 years ago? The answer would have to be no. So there's a lot of reasons for that, but it's certainly not prevalence. There are many, 1% of the population has schizophrenia, and more than that has Alzheimer's disease. So I think what the lesson, so next door to me, I should tell you, at the time, in the lab next door in the same department, at Merck, was an investigator named Costa Petruchin, who was working on monogenic forms of age-related macular degeneration, Stargardt's disease, Best macular dystrophy, those kinds of things. And he had a paper in Nature Genetics like every other week. And, you know, after like 3 years, I had one paper in BBRC, right? And so it showed me that not only can you make headway much more quickly in a rare disease, but that he was able to get the patient benefit, diagnostics, and at least biomarkers and prognosis, if not treatment. That's still been a ways coming, but gene therapy is in those diseases now. And so we tried to take a rare disease approach to schizophrenia, but there just are not naturally occurring forms of those kinds of disorders. There are an Alzheimer's disease, of course, but even translating that to the common has been difficult. So I think I would go back to what Harvey said, that is the famous Dr. Harvey, that is in the 1600s. If you look back at this famous quote that he said, this has been known for years, that if you want to understand how Mother Nature works, look at rare diseases, because they will tell you these are experiments of nature, if you will, but they are diseases which are of tremendous medical need, are more tractable, frankly, than the common ones, can teach us a lot of things about common diseases that are very hard to learn via common diseases. And I firmly believe, and you look at Alzheimer's disease, a great example, that by studying rare diseases, we will have an impact on the common diseases. And so that's why I would stay in the rare disease world. I also think that the distinction is becoming less and less relevant, like we got to before. These worlds are the common diseases are becoming segmented. The rare diseases, as we network the world, we're finding that the prevalence of these disorders, almost every disorder, is more prevalent than all of us were taught they are. And that's not only because we're able to look increasingly at national and international populations, but we're also not able to we don't just have to look at an extreme phenotype, right, which the diagnosis or the syndromic definition is always made on the basis of an extreme phenotype. If we look at less extreme phenotypes in case of different alleles, it'd be 10 times as common. You look at, my favorite example is multiple sclerosis. When I was in training as a neurology resident, MS was a rare disease. It's a common disease now. And it's not more common, it's just that we're better at diagnosing it.
That is pretty elegant. That was very nice. I like that. I saw that someone raised their hand, so if you want to yell at your question, please do.
Hi, this is Reena Kartha. I'm calling from the University of Minnesota.
Yeah, go ahead.
And I had a question regarding the tissue chip model. This has been a wonderful presentation, a great vision for the future, Dr. Austin.
Thank you.
But my question is about the tissue chip. So if you identify a compound and using the tissue chip, I'm assuming that FDA will still require the investigator to do some preclinical studies and animal models. Is that right?
Yes. And we're in, and this is something you can all help with, because this is a cultural change as well as anything else, and we need to help our colleagues at FDA, because they're trying to do the right thing, but they're really concerned about protecting public health. You know, it's first do no harm. So they're always, until we can demonstrate to them that the tissue chips really are more predictive, they're going to make us continue to do animals. So they're looking for solid science. Okay, so what's the real answer to your question? Yes, they are very intrigued by these systems. They don't want to have large numbers of animals used, put to death, et cetera, any more than any of us do, particularly if they're not predictive of human health and are not actually protecting public health. The reason that we put them on the project team from the beginning was that we wanted their input on what is sufficient data, what are sufficient data to allow the agency, allow FDA, and I was about to say EPA is interested in this too for the same kind of reason, just on environmental chemicals. What would represent a sufficient data set that we could begin to think about initially substituting an animal species with a tissue chip that is currently, you're probably aware, if you want to put an animal, put a drug in humans, you've got to do two species, usually a small animal and a large animal. That means mouse, rat, guinea pig, rabbit. On the small side, dog usually, or monkey, non-human primate. On the big side, pig, sheep sometimes. And so the question that we're now engaged with them is, at what point would you feel like the data looking at historically, retrospectively now, at compounds which either were safe or not safe in animals and humans, those retrospective data, at what point would you be willing to prospectively say that now I am not going to require you for this next project to do 2 animal species, I'm going to make you just do 1 and the chip. So then the next step would be, eliminated completely. I would say we're still a good year off, maybe probably 2, because I think it might happen in 1 year. It's probably 2 or 3 years realistically before it happens. But I must say, this program has the velocity of this program is faster than I thought it would be. So I could be underestimating here.
And would this model be good for testing gene therapy products?
Yes, it should be. I mean, because you figure if you just give you an example of how this works, a very concrete example, there's a current 3-organ system where this is an oral drug we're testing now, goes in an artificial oral fluid, goes into the artificial alimentary tract, goes into the artificial small intestine, gets absorbed out of the artificial small intestine, then goes to the artificial liver and gets metabolized, and then goes to the kidney chip, and in this case causes good things or bad things to happen in the kidney tubules. Now, there's no, you have all the same issues that you have with gene therapy in this kind of model. That is, could you be able to target these things, or is the vector, is the virus, whatever the vector is it going to recognize a cell type in just the target organ or an organ on the way there? There are blood vessel models here, so you can model doing IV treatment as well. So no, that it's and you should know that, interestingly, you might ask, well, I understand why NIH is interested in this. Why on earth is DARPA interested in this?
Mm-hmm.
And so I asked them that question at the beginning, 5 years ago. What's the matter with you people? Aren't you out, making airplanes and stuff? And they said, yes, the reason is that they got interested in this problem because during the Gulf War, soldiers were and are still exposed to really nasty pathogens and sometimes biowarfare agents that require countermeasures. And those are either drugs or they're vaccines or in some cases might be gene therapy too. And so they were very motivated to develop a testing system that would be agnostic. So what I would say is that where we are in this process now is a really exciting time where there are prototypes of all of these. But if you've ever known a technology developer, and this is true with software developers, it's true with engineers, who have a new gadget, they just can't wait for you to take this out for a spin and see if you can get it to work. And it almost never does, and you end up working with them to make it optimal. But lots of these people are eager to work with investigators who have systems which they could use or collaborate with you on to find out what the scope of use is for these. I wouldn't approach it, if you're interested in doing this, I wouldn't approach it as, Dr. Jones with the liver chip has a plug-and-play answer, because he's not at this point. It would really be a collaboration to investigate the utility.
Okay. Thank you.
Thank you. Anyone else? Okay. Well, I'm going to wrap us up. Thank you so much, everyone who could join us live. And if you're watching us after the fact, thanks for watching us this evening. Have a great day, everyone, and don't forget your January calls. I think we just added a new topic. Maybe talk about if you could somehow come up with a use for the cell system that you could think about using in your work on the January call. So start brainstorming and be creative. Thank you, Dr. Austin, and thanks, everyone, for calling in. Have a good afternoon, everyone. Bye.
Thank you.
Thank you, Dr. Austin.